Hello students, welcome to chapter 8. We're going to be talking about photosynthesis. Keep in mind that photosynthesis itself doesn't only happen in plants, it also happens in lower life forms. We talked about bacteria that has chlorophyll in it that can get sunlight, that they can get that sunlight energy and convert that into chemical energy. But mostly we will be talking about plants because they are eukaryotic, that they can have the chloroplasts and everything inside of them that have the chlorophyll. Light reactions involve two main processes. They have to absorb the light, and they have to have the energy transferring molecules, and they have to have the energy absorbing molecules, such as NADPH and ATP, right? So we have the electromagnetic spectrum. We have that visible light. So it starts from purple and goes to dark red. Forms of radiant energy differ in wavelength. So we start from a little bit below 400 nanometers to about 800 nanometers. That is our visible light spectrum. Horizontal distance between these crests, that's our wavelength. Okay, so wavelength is that horizontal distance between those crests of light, those wavelengths. And then you have frequency, which is however many waves go in between a certain amount of distance. Visible light has wavelengths between about 700, that goes a little bit beyond that, and about 400, which is blue light, that goes a little bit below that. We can see the entire spectrum combined together as white light. It's just a spectrum of what you call visible light. So that's our visible spectrum. And white light itself doesn't refer to that whole spectrum, because there is spots where you can't see in between that spectrum, because your eyes don't catch certain spectrums in between that visible light. It's a complicated measure, but we're not going to get into that. Energy is using energy in a unit of light. So light itself is photons whenever you see it. So light itself is a wave, but it's also photons. Is inversely proportional to its wavelength. So light acts as a wavelength, as a wave, and it also acts as a material. It acts as a photon. That's its physical embodiment. So the shorter the wavelength, the greater the energy of the photon. So as you get down into the 400 range, as you even get down into the ultraviolet, X-rays, gamma rays, more energy is released from those photons. But as you get higher into the wavelengths, as you get into the radar, infrarays, TV rays, shorter wavelengths, or the short waves, those photons have lower energies. Physics of light energy, this is where the photons come into play. So when not absorbing light, electrons in a pigment molecule are at relatively low energy state. So when you have an atom that center molecule is your core, right? You have the protons and neutrons in the center, right? But on the outside, you have that electron cloud. So a photon will hit the electron at its ground state and excite that electron. So when an electron in a pigment absorbs the energy of a photon, it jumps to a higher state, right? So electron is here, and a photon comes in and it hits it, and it jumps to a higher state. It jumps to a higher cloud. The difference in energy level between its ground state and an excited state depends on the energy of the photon absorbed, right? So let's say you have a photon here, electron here, and a photon comes through and it's at a really high energy. And it comes in, it hits that electron, and it bounces it really high. Well, it needs to come back down. It's not going to go up and then just stay there. It'll come back down, right? Whatever comes up will come back down. Whenever it comes back down, that photon will be released again. So it'll hit it, it'll come back up, and it'll come back down. That photon will be released as a color or as light. Most of the time, it'll release it as both. So it'll release it as electron, or it'll release it as a pigment. Okay, the excited higher energy molecule from the pigment returns its ground to its ground state by emitting light of a longer wavelength, right? Because it's absorbing some of that energy. So it's releasing it at a longer wavelength than the absorbed light, or releasing its energy as heat, right? It'll release it as heat, some type of heat transfer. The excited electron is transferred from the pigment to a nearby electron accepting molecule, right? That's NAD, NADPH, something. The energy of the excited electron, but not the electron itself, is transferred to a neighboring pigment molecule. The pigment molecule, in this case, being chlorophyll. This transfer excites the second molecule while the first molecule returns to its ground state. Very little energy is lost in this transfer of energy. So that energy is transferred to the chlorophyll, thus releasing some green color, right? So a photon comes in, some energy is released as heat. That electron accepting molecule is accepting the electron from some matter, and then some energy is transferred to the chlorophyll. Absorption spectrum and action spectrum. If you ever wonder why it's green, this is going to be why. Light absorbed by the carotenoids and chlorophylls, so carotenoids, carrot, meaning orange, and chlorophylls meaning the green, chloro meaning green, phylls meaning leaf. Acting in combination drives the reactions of photosynthesis. Green in this state is the lowest form of energy, so they don't really want to keep that energy, they want to release that energy. So the amount of light of different wavelengths that is absorbed by the pigment is an absorption spectrum. Okay, every other color here is absorbed, right? So we have blue and violet, and then red on the other side. So blue and violet is a very high energy color. It's a very high energy wavelength, right? And then orange and red is also somewhat high. Green isn't really high. Green and yellow, that's not really a high energy, okay? But blue and violet is a very high energy, so that's what they're going to gain the most of. And then red and orange, they're going to gain a little bit of that. The effectiveness of light of each wavelength in driving photosynthesis produces a graph called an action spectrum. Okay, so action spectrum is where all of their photosynthesis is going to take place. Whatever spectrum that they can get the most action out of in photosynthesis, that's where they're going to get the most amount of energy from. So you can see the absorption spectrum and the action spectrum are both acting the same area of wavelength on the spectrum lines. Also, in the physics of light energy, we have where the excited electron returns to its ground state, right? It releases energy as heat or light. We kind of talked about this before, right? And this is known as fluorescence. Some type of light, some type of heat is just released, right? That's always the case. Whenever you have a transfer of energy from one state to another, as we talked about this before, it's going to be released as heat. The excited electron is transferred from the pigment molecule to a nearby accepting molecule. So whatever pigment it's going to be accepting that molecule, that's its primary acceptor, right? And it's often going to be NADPH, right? So NADP is going to be accepting that H. Just like we said in cellular respiration, it's going to be accepting that H involved. But in this case, it has an extra phosphate in it. Or an extra phosphorus. So photosynthesis has an extra phosphorus in it. Cellular respiration doesn't. Photosynthetic pigments absorb photons and transfer excited electrons to the primary acceptor molecules. So it just excites those electrons, right? And then accepts those electrons to that primary acceptor molecule. That H comes in and binds to that NADP. And that H carries those electrons. The energy of the excited electron is transferred to a neighboring pigment molecule, and the first molecule returns to its ground state. So this is rather ingenious. Basically what happens is each pigment carries the next molecule, carries the next energy to the next molecule. So the pigment molecule will carry the next pigment molecule, will carry the next pigment molecule. All the energy goes to the next pigment molecule. So it doesn't allow that energy to remain, it allows that energy to remain within the system until it essentially goes away. So it'll absorb as much energy as it can until it dissipates into nothing. It's a conservation of energy until it can suck as much energy out as it can until it eventually can't do anything with it. There's an experiment that Theodore Engelmann used to try and figure out what colors within the visible light spectrum that these types of chlorophyll used. And he used a glass prism to break apart white light or sunlight or whatever. He broke apart light into a spectrum of colors, which is cast across a microscope slide with a strand of algae and aerobic bacteria. So this algae was photosynthetic, right? And he saw that bacteria, this photosynthetic bacteria, grew at best where there was blue and violet light, and grew at best where there was some red light, right? And we see this in this image right here. And we know this because the visible areas on green algae, it emits green light, right? So it's not going to try and absorb that green light. It's going to try and absorb other colors of light. So you think you would know that, but he wanted to do an experiment where he saw, well, what types of light, what types of wavelengths do these bacteria use the most of? What types of light do they thrive in? In photosynthesis, light is absorbed by green pigments, right? And that's in chlorophyll. But there's also other colors that you see in plants, right? So it's not all green. You see bell peppers that have the orange pigments. You see flowers that have purple pigments. So yellow-orange pigments come from carotenoids, anthocyanins and betalains. Those all have kind of a reddish color. Betalains especially, they have the reddish color, and beta comes from the word, beta can also be synonymous with beets. And if you ever chop a beet in half, you can see it's red, right? Anthocyanins can either be red or they can be purplish. Anthocyanins especially are in peaches and plums. And if you ever chopped open a plum before, it's going to be like a purplish color. So those anthocyanins keep those purplish colors. Like in blueberries, that have a lot of anthocyanins in it. Things like bananas, they'll have xanthophylls. So what are those useful for, right? So this pigment color is produced by transmitting or reflecting different colors of light, right? So if you saw the action spectrum or the absorption spectrum of those, you would see that those lines go in a different pattern. So plants look green because chlorophyll absorbs blue and red light. Well, if you saw the anthocyanins, they would absorb other colors like green light, or they would absorb more red light or other colors. So on these, they're more useful for attracting things to eat them, right? So when you eat them, they're going to go through your system and the seeds are going to come out the end. So when they come out the end, they fertilize in the ground and, they come out as trees or bushes or other plants. When you have a flower, it's going to attract pollinators. So they're more useful for attracting animals. And if you see these types of colors, they're going to be in fruits, they're going to be in flowers, they're going to be in the reproductive parts of the plants, rather than on the leafy greens. Autotrophs and heterotrophs. So here we're going to talk about more of the ecological side of plants, and the more side where chlorophyll is based in why they need chlorophyll. So autotrophs, we know what autotrophs are. Autotrophic, self-eating, self-fulfilling. So autotrophs, organisms that require organic molecules from inorganic sources such as CO2 and water. They don't really need sunlight, right? They just need the ground in order to survive, and that's it. They can get most of the materials from other means. There are other things that it can get it from. Autotrophs, these are things like mushrooms. Photoautotrophs, these are things that require light. So these are autotrophs that use light as an energy source. So just the word photo in front of autotrophs. Photo meaning light, autotroph in front of it, or in back of it. So photoautotrophs, yeah. And then you have heterotrophs. So there are plants that are strictly heterotrophs. Consumers and decomposers. So these are plants that actually use what's called a hostorium. And they will dig into another plant, dig into its root, and suck all of its energy out of that other plant. So this is useful in many types of plants, and they will actually not have any green leaves at all. A lot of these plants will actually be flowers, just flowers. And they will open up at a certain time of the year and die off once it's produced fruits, and then go back into the ground or go back into the tree or plant that it's been infecting. So two kind of examples here. Mistletoe is actually called a photoheterotroph. It can survive on its own, but it mostly has those leaves in order to photosynthesize if the tree isn't doing well where it is taking its life from. So the mistletoe will infect the tree, take its moisture, take its sugars from the tree where it's hosting from, and then grow from there. And actually birds spread mistletoe. This is the mistletoe historically from the pagan Christmas that it is actually from. And then there's another one called conophytum. Conophytum, this is a plant that's in the ground, and it'll hostorium into plant roots, into tree roots, and it'll live in the ground until a certain time of year, and then it'll pop up with flowers. Little flies will go into the flowers, pollinate it. It'll spread its seed, and then it'll go back into the ground. As your attendance, I want you to give me another species of plant heterotroph. Just look online, give me a citation from where you found that species, and then that'll be it. Just give me a species, give me the genus and the epithet of that species. It's pretty easy. You can give me a heterotroph, you can give me a photoheterotroph, give me something else other than the ones that I've given you. Okay? And of course, give me the citation. All right? Okay. So, movement of materials throughout the plant. CO2 enters the plant through the stomata. So on the leaf, there's little holes on the bottom of the leaf, sometimes on the top. Little holes on the bottom of the leaf where CO2 enters and then diffuses into the cells of the leaf, right? So that is useful for photosynthesis. O2 and water produced in photosynthesis diffuse out through the cells and transpire through water vapor and O2 through the stomata as well. So O2 and H2O come out and CO2 enters in through that stomata. Also, water and minerals are absorbed by the roots. So comes in through the roots, right? Comes in through the roots, comes up into the stem and up into the leaves. Those are transported to the cells through these tubular conductive vessels. Okay, so you have the xylem and the phloem. The xylem transports water, and the phloem transports sugars down into the roots. So the xylem transports water up, the phloem transports sugar down. Chloroplasts are formed from 3 membranes that enclose 3 compartments. Mostly we have the outer membrane, we have the inner membrane, and the thylakoid membranes inside. The compartment formed by the inner membrane, so we have the outer membrane, inner membrane, inside that is the stroma. The stroma is the fluid inside. Within the stroma, we have the thylakoid membranes, right? So in that sac, in that fluid, is those thylakoid membranes. So the thylakoid membranes are arranged in stacks. Those stacks, all those stacks together, are grana. Okay, grana, like different grana, and then if you have one stack, it's a granum. It's kind of weird, but you know. So grana are interconnected by flattened tubular membranes called lamella. So they're just interconnected by a lamella. And then stromal lamella probably link thylakoid lumens to a single continuous space. So it's just all connected together. So now we're going to talk about photosynthesis itself. Okay. First, we have what's called the light reactions. These are just what's happening when you have the energy of the sun coming in. So we talked about this somewhat when we talked about what's happening when the light is actually shining whenever it's bouncing from one pigment to another. So that's converting into chemical energy. That's ATP and NADPH, right? NADPH carries those electrons pushed to a higher energy level and by absorbing that light, right? So we have the higher energy level carrying that NADPH, carrying that electrons from one, energy to another. This all takes place within the thylakoid membrane. The thylakoid membrane is what's carrying those pigment molecules. After that, we have the Calvin cycle. And the Calvin cycle uses those NADPH as a source of energy to convert CO2 into an organic form, and this is called CO2 fixation. All right, CO2 fixation is a reduction reaction in which the electrons and protons are added to CO2. So you have the CO2 molecule coming in where the NADPH right, is added to that, right? The NADP goes off, gets more H's, but it's added to CO2 to make sugars. That's fixed, right? And that's basically making sugars for more energy to be put into cellular respiration. All right, so we have CO2 plus H plus electrons goes into that sugar molecule. So we have oxygen from hydrolysis. Certain experiments that have been done using different isotopes show where each atom of reactants from photosynthesis end up in the products. So water coming from the roots comes up, and that makes oxygen. CO2 coming from outside going through into the stroma makes that sugar. Also, that oxygen from the CO2 makes more water, and that comes out through the stroma. It's a little unintuitive to know that water comes up through the roots and then makes oxygen, but also it comes out through the reaction. But cellular respiration kind of does the same thing. We need water, and then we also make water. So it's a little give and take situation, and also photosynthesis does the same thing. So oxygen released by hydrolysis or water splitting, hydrolysis, water splitting. So water splitting reaction photosynthesis profoundly changed the Earth's atmosphere. Okay, so what does it mean by that? Whenever the Earth was starting to form, we didn't have oxygen. Okay? Photosynthesis started to take place in what's known as pre-algae. Okay? So these pre-algae forms started to form hydrolysis, right? So water started to split in these pre-algae forms and started to make oxygen. All that oxygen started to rise in the atmosphere and started to make this oxygenated atmosphere and started to build what's known as a chemical reaction in the atmosphere. Now, it's highly conjectured, but scientists believe that photosynthesis formed before, cellular respiration, because of this reason. Cellular respiration could not have formed in an oxygen-low environment. So photosynthesis had to have formed first because of this water splitting molecule, this hydrolysis event. Let's talk about the light reactions in a little bit more detail. So the light reactions involve two different systems, and then what we know as the ATP synthase. So photosystem 2 starts first, and then photosystem 1. And this was just because photosystem 2 was found after photosystem 1. It's a little bit complicated, but you have to realize that photosystem 1 was found first, and then they found photosystem 2, and they couldn't mix up the words. So, first protein complex in the light reactions of oxygenic photosynthesis. So light comes in, and then water comes in to split into oxygen and hydrogen. So oxygen is the first byproduct of photosynthesis. That hydrogen is split off and taken off into the inner thylakoid lumen. So light splits that hydrogen and oxygen. That's hydrolysis. And that happens in photosystem 2. And this all happens at this optimum wavelength, right? That optimum wavelength at P 680 at photosystem 2. Then it goes through a cycle of plastoquinone, right? That hydrogen comes in, builds that plastoquinone, and plastoquinone goes through, builds it up, and then another hydrogen comes in and goes into the thylakoid lumen. Goes into the cytochrome, right? That's right into there. Reduces plastoquinone into plastoquinol. The energized electrons are replaced by oxidizing water to form hydrogen and oxygen. Then we have photosystem 1, the second protein complex in the light reactions. Protein complex, it means that there's multiples. So protein complex in the light reactions catalyzes the transfer of electrons across the thylakoid membrane from plastocyanin to ferroxydoxin. Ferredoxins. So the plastocyanin that came from that cytochrome goes into that photosystem 1 and then used to produce the high energy carrier NADPH. So NADP comes in, binds on to ferredoxin. Ferredoxin gives it a hydrogen. NADPH is carried off. So that NADPH goes off, and then you're basically done. So photosystems are the sites at which light energy is converted into chemical energy. So all that chemical energy is created. But all these hydrogens that are coming in have to go somewhere. So that thylakoid lumen, that thylakoid membrane, is being filled up with hydrogens again. Just like we saw in the cellular respiration. So those hydrogens go through the ATP synthase. And if you saw before, that's chemiosmosis, right? It's that proton pump. That's a proton force. So hydrogens go up through the ATP synthase to give it more ATP. Each photosystem consists of an antenna complex and a reaction center. So that antenna complex just tells it what it needs to do, and then reaction center is where it needs to react to tell it the optimum wavelength at where it needs to go. So photosystem 2 has an optimum wavelength of P680, and photosystem 1 has an optimum wavelength of 700. So electrons from water first flow through the photosystem 1, becoming excited to a higher energy level through first absorbing light energy at P680. So it goes up, all those electrons go up at 680, And then the electrons go downhill in an energy level through electron transfer system, connecting 2 and 1. That's through the cytochrome, through all those plastic quinones and everything. Through photosystem 1, they're excited a second time through absorbed and light energy at P700. High energy electrons pass through an electron transfer system consisting of a series of high energy carriers: plastoquinone, cytochrome complex, and the mobile carrier through plastocyanin. So they're ultimately reduced and oxidized, releasing free energy at each transfer. So all of that is releasing free energy all the time. Some of this free energy is used to create a gradient of hydrogen across that membrane. So it's releasing hydrogen across the membrane, giving that hydrogen over to the ATP synthase. So then we get to the ATP synthase, just like that in mitochondria, and that gradient of protons allows for the production of ATP through ADP and phosphorus, or phosphate, through proton motive force, through chemoosmosis. This is more of a detailed picture of that. So synthesis of ATP, this is photophosphorylation because you're adding light to this reaction. So synthesis of ATP coupled by the transfer of electrons, energy through photons of light. After all of that, what we have is CO2 coming in. This is carbon fixation. We have this NADPH at the end. And that NADPH has to go somewhere. NADPH, we have ATP, we have light energy that has gone through, and then we have CO2. So CO2 is added to ribulose 1,5-biphosphate, and this is RUBP. So CO2 is coming in, right? That's added to what's called the reactants of the first Calvin cycle. A transient 6 carbon molecule is cleaved to produce 2 3 carbon molecules. So you have 2 3 carbon molecules that split into 2 3 carbon molecules that's 3-PGA. Some ATP goes in, some NADPH goes in to produce basically G3P. That basically makes 1 carbon molecule. Okay, it takes a full rotation to make 1 carbon molecule at a time. At the end of it all, you have to make 6 rotations to make 1 glucose molecule. And it's kind of difficult to think about that, but this whole rotation makes 1 carbon molecule at a time per go round. So plants that initially fix carbon in this way are C3 plants. So a phosphate group from ATP and electrons from NADPH are transferred to 3-PGA and produce 2,3-G3P, right? After 3 turns, 1 G3P exits the cycle. That's what makes it C3, right? Because that's carbon free. 5 G3P are used to generate ribisco biphosphate, or what's called rubisco. Ribulose 3-biphosphate. So you have to make that over and over and over again just to make one glucose molecule. Once G3P enters a complex series of reactions that yields the 5-carbon sugar, it can go back into the reaction and finally make a sucrose or starch. In the final reaction of the cycle, a phosphate group is transferred from ATP to generate the ribulose 5-biphosphate used in the first reaction. So for each turn, one CO2 is converted into one carbon hydrogen to oxygen unit. So you have one turn, each CO2 is converted into one carbon unit. It takes 3 turns just to make a net 3G phosphate. So net 3GP is primary building block for any reaction producing glucose and many other organic molecules in chloroplasts. It takes 6 turns just to make glucose. So here's the final reactant equation. You have CO2, 2 NADPH plus 3 ATP, producing 1 CH2O plus 2 NADP plus 3 ADP plus 3 phosphate. Photorespiration. Photorespiration happens when you have too much oxygen in one area and not enough CO2. Okay? This binds oxygen and releases CO2. When oxygen binds to rubisco, RUBP is converted into 3-PGA and phosphoglycolate, basically meaning that no carbon is fixed. All right, phosphoglycolate is broken down into glycolate, which is oxidized into CO2. You have this carbon that is lost in the reaction, and no ATP is produced. Now that's bad for the plant because it doesn't produce energy. It doesn't produce any type of oxygen or any type of energy in the reaction. Photorespiration is also reducing efficiency of photosynthesis by removing some intermediates of the Calvin cycle. Plants in very hot, dry climates. So in hot, dry climates, you have very little CO2, you have very little amounts of animals or anything that produces CO2. So they reduce the amount of their open stomata in daylight to conserve water. And they also conserve the amount of energy needed to open their stomata and let in CO2. So during the night, the CO2 actually drops in the atmosphere whenever it's cold. Plants open or close the stomata to balance the demands for gas exchange or water loss. And then you have alternative fixation. So there's alternative ways in order for plants to handle that type of photorespiration. Some plant species in these hot, dry environments use what's called a C4 pathway. So normal plants have a C3 pathway. This is a C4 pathway. CO2 is fixed as oxaloacetate. This is CO2 and phosphophenol pyruvate, or PEP, as a carbon 4 molecule of 4-carbon molecule. So what they'll do is in the mesophyll cell, they'll fix it as an oxaloacetate, right? And then they'll also convert it into a malate or malic acid. And they'll store that in the bundle sheath cell. Okay, so whenever they need that that type of carbon, they'll just pull it out of malate. So they'll pull it out of malate, convert that back into CO2, and then it can go into the Calvin cycle whenever it needs it. So it doesn't have to open up its stomata in order to get CO2, it can just pull it out of malate. These types of C4 plants have what's called a Kranz anatomy. So in other types of plants, they have a normal type of anatomy, but these have a Kranz anatomy where they have larger bundle sheath cells, they'll have larger mesophyll cells in order to store that malate in their vacuoles. So C4 plants are much more efficient at fixing C3 or CO2 than C3 plants. But more ATP is required in order to do that because they have to pull it out of that malic acid. They have to pull it, basically convert that malic acid back into CO2. C4 plants include crabgrass, corn, sugarcane, things that are grass-type plants. These are C4 plants. And then we have other types of plants that kind of follow the same pathways as C4. So these are CAM plants. But these are temporal fixation. So in temporal, I mean carbon fixation occurs at night, and then the Calvin cycle occurs during the day. So these have, again, special proteins such as malic dehydrogenase to convert that malic acid back into CO2 in order for it to go through its Calvin cycle again. So it can convert that back into malic acid to be used during the day during the carbon fixation reactions. But instead of just to be needed, it does it during the day, and then it'll absorb CO2 at night. So CAM pathways are more temporal, and then C4 pathways are more physical barriers. CAM plants are things like cactus, orchid families, lily families. So the lily family are things like the pineapple, the Spanish moss, crassula, crassulation, acid metabolism, crassula. This is the emerald or the jade plant, but orchids are also CAM plants. Stomata on fleshy leaves, they open during the night and then they close during the day due to water loss. Okay, make sure to find the attendance assignment, make sure to do that, then make sure to go through the lab for today. Okay, and then I will see you later.